Media Advisory, Wednesday, March 29, 2023. Strategy may speed delivery of therapeutic options relative to time it would take to develop gene therapies. A National Institutes of Health team has identified a compound already approved by the U.S. Food and Drug Administration that keeps light-sensitive photoreceptors alive in 3 models of Leber congenital amaurosis type 10 LCA10, an inherited retinal ciliopathy disease that often results in severe visual impairment or blindness in early childhood. LCA10 is caused by mutations of the cilia centrosomal gene C, EP290. Such mutations account for 20% to 25% of all LCA more than any other gene. In addition to LCA-C, EP290 mutations can cause multiple syndromic diseases involving a range of organ systems. Using a mouse model of LC8 and 2 types of lab-created tissues from stem cells known as organoids, the team screened more than 6,000 FDA-approved compounds to identify ones that promoted survival of photoreceptors, the types of cells that die in LCA, leading to vision loss. The high-throughput screening identified 5 potential drug candidates, including reserpine, an old medication previously used to treat high blood pressure. Observation of the LCA models treated with reserpine shed light on the underlying biology of retinal ciliopathies, suggesting new targets for future exploration. Specifically, the models showed a dysregulation of autophagy, the process by which cells break down old or abnormal proteins, which in this case resulted in abnormal primary cilia, a microtubule organelle that protrudes from the surface of most cell types. In LC8 and C, EP290 gene mutations cause dysfunction of the primary cilium in retinal cells. Reserpine appeared to partially restore autophagy, resulting in improved primary cilium assembly. Reserpine targets dysregulated intracellular signaling pathways downstream of the primary cilium. Such a treatment strategy could potentially address retinal ciliopathies caused by many of the more than 160 disease-causing genes, regardless of the specific gene involved. That's in contrast to gene therapy, which requires a very expensive and labor-intensive process to tailor an individual gene-based therapeutic approach for each mutation. This work was supported by the NEI Intramural Research Program ZIA-IF 150 and ZIA-IF 146 and the National Center for Advancing Translational Sciences Intramural Research Program ZIA-18-6. Anand Swarup, Ph.D., Senior Investigator and Chief of the NEI Neurobiology Neurogeneration and Repair Laboratory. To schedule interviews with Dr. Swarup, contact NEI at nenews@ne.gov. Shanghai Swarup M., Petal S. Mondal AK Song HB, Camp LOL, Tawa JJ, Region F, Shimada H. Nagashima K, Deval N. Jacobson SG, Jing W. Swaruba. Reserpine maintains photoreceptor survival in retinal ciliopathy by resolving protostoses, imbalance and ciliogenesis defects. eLife March 28, 2023, https://doi.org/10.7554/eLife83205. About the NEI, NEI leads the federal government's efforts to eliminate vision loss and improve quality of life through vision research, driving innovation, fostering collaboration, expanding the vision workforce, and educating the public and key stakeholders. NEI supports basic and clinical science programs to develop sight-saving treatments and to broaden opportunities for people with vision impairment. For more information, visit https://www.nene.gov. About the National Institutes of Health. National Institute of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers, and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research, and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health Turning Discovery into Health Registered.